Hi, my name is Rachel Silvio, and my topic for the individual topic video presentation for Bio 525 is Toxoplasmosis. The objective of this lesson is that students will be able to understand the structure, pathogenesis, epidemiology, treatment, and prevention of toxoplasmosis caused by the parasite Toxoplasma gondii, seen here in the background picture of this slide. Toxoplasmosis is a disease of the blood and lymphatic vessels. It is caused by the protozoan Toxoplasma gondii. This protozoan, like the malarial parasite Plasmodium, forms spores. Tigondii was first discovered in 1908 by Charles Nicole and Louis Monch after they found the parasites in the blood, spleen, and liver of Tigondii gondii, a North American rodent seen in this picture. Alfonso Splendore also discovered the parasite the same year in a rabbit in Brazil. It was named a year later in 1909. Toxoplasma means arc-like form, and the name gondii is after the rodent it was discovered in. Tigondii was found in tissues of a congenitally infected infant in 1939. In 1948, researchers identified Tigondii as a parasite of warm-blooded hosts around the world using a Sabine-Feldman dye test, which is a specific antibody test. Its life cycle was discovered in 1970, helping us learn that cats are the definitive host and infection can be caused by contact with parasitic oocysts in the cat's feces. Here is the classification of this parasite. Tigondii is a protist in the alveolate superphylum with ciliates such as Paramecium and dinoflagellates such as red algae. It is in the phylum Apicomplexa, along with Plasmodium, the parasite that causes malaria. There is only one species, Gondii, within the genus Toxoplasma. There are several strains within that species seen around the world. Cats are the definitive hosts of Tigondii as the parasite undergoes its sexual phase in the intestinal tract of the cat without causing any symptoms in the cats. Millions of oocysts are shed into the cat's waste products for 7 to 21 days after infection, contaminating food or water ingested by other animals with immature oocysts. Sporogony, asexual reproduction, occurs, developing the immature oocysts into mature oocysts, which contain 2 sporocysts, each with 4 infective sporozoites. Oocysts then infect the intermediate hosts, such as mice or other rodents, domestic animals, such as sheep or cows, and humans. If rodents are infected, they lose their normal avoidance behavior of cats, making them even more likely to infect cats. If domestic animals are infected, such as beef cows, then humans can become infected if they eat undercooked meat containing tissue cysts. Once they have been infected as an intermediate host, the sporozoites invade the host cells and form trophozoites called tachyzoites. A rapid reproduction of the tachyzoites causes the host cell to rupture, releasing more tachyzoites, causing a strong inflammatory response in the host. As the host's immune system fights the parasite, the infected host cells develop a wall, forming a tissue cyst. The parasites in the cyst reproduce slowly, and this stage of the infection, called bredizoites, may persist for years, especially in the brain. These bradyzoites are what the cats ingest by eating infected mice, and the cycle begins again. Tigondii is a eukaryotic unicellular organism. There are 3 infectious stages of Tigondii: the tachyozoites in groups, the bradyzoites in tissue cysts, and the sporozoites in oocysts. When this protozoan is in the tachyozoite stage, they are about the size of large bacteria, 2 by 7 micrometers. The picture on the left shows a drawn diagram of the tachyozoite and a bradyzoite. The picture on the right is an electron micrograph of a tachyozoite taken from a mouse. This electron micrograph on the left shows a mouse macrophage with several tachyozoites. You can see on the top of the image a tachyozoite escaping the macrophage, with the arrow pointing to it. On the right picture, you get a better idea of how this tachyozoite escapes the macrophage. Here is a negatively stained apical complex unique to the phylum Apicomplexa, with rings made from microtubules on the tapered end of the cell that assists the cell with invading host cells. Those with healthy immune systems show mild symptoms, or none at all, if infected with Toxoplasma gondii. Between 22 to 40% of the human population over the age of 26 will develop antibodies to this microbe, most without even knowing it. Infection rates are different for different countries. 7% in the UK, 11% in the US, and 67% in Brazil. Now, the 2 most common ways of humans becoming infected is eating undercooked meat with the parasite, or by direct contact with cat waste products, where the parasite is located, usually by cleaning out a litter box. A study done by researchers from the National Institute of Health focused on the geographic component of the variation of the parasite, discovering that most populations of Tigondii are specific to location, but some populations are found worldwide. Tigondii is unique because they found one population in all continents except South America, and another population was found only in South America. Now, evidence indicated that South American and Eurasian populations evolved separately. And it wasn't until recent evolutionary history that the 2 strains were introduced to one another when rodents and cats migrated on ships during the transatlantic slave trade. Another interesting characteristic of this species is, despite its ability to infect almost all mammal and bird populations, the genetic diversity of this parasite is surprisingly low. There may be mild flu-like symptoms in the first few weeks of infection, but most people do not have any symptoms at all. In individuals with weakened immune systems, there may be severe symptoms. Some researchers have linked infection of this parasite to personality changes, mental illnesses, such as schizophrenia, anxiety and bipolar disorder, increased risk of traffic accidents, and even changes in human culture. Some have even hypothesized that an infection by Tigondii could be linked to brain cancer. Tigondii infection associated with 1.8-fold increase in risk of brain cancers, although this might just be correlational. The parasite causes a cyst in the brain, provoking inflammation and inhibiting apoptosis, which is similar to brain cancer. Researchers found that patients with meningiomas were twice as likely to test positive for Tigondii as people without it. Yet more research must be done to find this link. The primary danger of infection with Tigondii is congenital infection of a fetus. An infection in the mother can cause a stillbirth or a child with severe brain damage or vision problems. It is estimated that there are 4,000 cases in the U.S. annually. Fetal damage occurs only when the woman acquires the initial infection during pregnancy. Some European countries encourage abortion if the woman becomes infected during pregnancy. Wildlife is also threatened by this parasite. Oocysts and wastewater contaminate freshwater runoff into the ocean. Researchers from the Morris Animal Foundation discovered that infected bobcats and red foxes were contaminating the nearby watersheds, causing concentrated sources of Tigondii in filter feeders such as mussels. California sea otters were eating the mussels, and it was causing brain infections and fatal encephalitis in them, an already threatened species. There is also the danger of opportunistic infection. Persons living with weakened immune systems, such as HIV patients, can become infected with toxoplasmosis as a secondary infection. A lower immune system function reactivates the protozoan from tissue cysts and can cause severe neurological impairments or damage vision. Healthcare professionals use serological tests to detect toxoplasmosis. Recently, PCR tests have become available that approach an accuracy of 100%. Available treatments are combination medications of pyrimethamine, which is also used as an anti-malarial medication, sulfadiazine, and folinic acid. Unfortunately, this treatment does not affect the chronic proteazoite stage and can be quite toxic to the patient. In order to prevent infection of Tigondii, they recommend that you ensure the meat is cooked to prevent ingesting tachyozoites of the parasite. It is also important that pregnant women not handle cat litter. Hygiene is the best preventative measure, so washing hands after touching cat litter or raw meat is the best way to prevent an infection. Researchers have also currently been developing a toxoplasmosis vaccine. Toxoplasma gondii uses alpha amylase to digest starch. The researchers created a mutation in the enzyme, and results were promising in the development of a successful toxoplasmosis vaccine. Several rates of immunized mice that were reinfected with tachyozoites of Toxoplasma gondii were nearly 100% for survival. The new vaccine could both eliminate the reproduction of the parasite in mice and prevent them from forming cysts. The goal is threefold, shown in the picture on the left of the slide. One is to prevent congenital disease. Two is to reduce the tissue cysts in animals that provide food for humans. And three, to vaccinate cats in order to prevent oocyst shedding in order to reduce contamination from cat feces.